Good morning students. We are going to discuss amino acids and proteins. First, let us discuss introduction. So what is the meaning of amino acids? Here 20% of the human body is made up of proteins. Proteins are the large complex molecules that are critical for normal functioning of cells. They are essential for the structure, function and regulation of the body tissues and organs. And next, what is the meaning of amino acids? Here, proteins are made up of smaller units called amino acids, which are building blocks of proteins. They are attached to one another by peptide bonds, forming a large long chain proteins. So here what is the meaning of peptide bonds? We are discussing in the next slide. And the next topic is amino acid structure and its classifications. So amino acids are based on the classification. It is first is an amino acid contain both the carboxylic groups. Here see in the structure carboxylic group COOH and amino group NH2. Amino acids that have an amino group bonded directly to the alpha carbon atom referred to as a alpha amino acids. Every alpha amino acids has a carbon atom called an alpha carbon bonded to carboxylic acid COOH group. And amino group is here NH2 group, hydrogen atom. A hydrogen atom and R group that is unique for every amino acids. Classification of amino acids. There are 20 amino acids based on the nature of the R group. They are classified based on their polarities. So amino acids. So amino acids is classified into hydrophobic amino acids and hydrophilic amino acids. Here it is subclassified into polar and non-polar. So first one non-polar. Non-polar here R is indicated by maybe in the alkyl group or aromatic group. So alkyl group examples of amino acids are glycine, aniline, valine, leucine, isoleucine, methionine, proline. R group, that is aromatic groups, examples for amino acids are phenylalanine, tryptophan. And the next one is hydrophilic amino acids. Here also it is polar, so that is neutral, acidic and basics. So neutral amino acids are trypsin, serine, threonine, cysteine, glutamine, asparagine. The acetic based amino acids are glutamic acid, aspartic acid, and next is basic based amino acids are lysine, histidine and arginine. These are the classification. The next one is another one classification is classification based on essentiality. Here it can be classified into two types. One is essential amino acids and the next one is non-essential amino acids. So essential amino acids are the amino acids which you need through your diet because your body cannot make them. That is essential amino acids. So what is the next? Non-essential amino acid, which is, whereas non-essential amino acids are the amino acids which are not essential part of your diet because they cannot be synthesized by your body. So now I hope you understand the difference between the essential and non-essential. So the next one is examples for essential and non-essential amino acids. So essential amino acids are histidine, isoleucine, leucine, methionine, phenylalanine, threonine, tryptophan, valine. These are the examples for essential amino acids. And next one is non-essential amino acids. Alanine, and next one is glutamic acid, glycine or theanine, proline, serine, thiosin. These are the examples for non-essential amino acids. Next one is peptide bond. Peptide bond is amino acids are linked together by amide group. So you know amide group is NH2 group, so called peptide groups. During proton synthesis, the carboxyl group COOH group of amino acids at the end of the growing polypeptide chain react with the amino group of an incoming amino acids, releasing a molecule of water. The resulting bond between the amino acid is a peptide bond. See in the below structure, here one of the oxygen and two molecules of the amide group hydrogen molecule is found into the water and it is released. So the next structure, the peptide bond is formed after removing of hydrogen molecule. Next one is important, structure of proteins. The sequence of a protein is determined by the DNA of the gene that encodes the proteins. A change in the gene's DNA sequence may lead to a change in the amino acid sequence of the protein. Even changing just one amino acid in a protein sequence can affect the protein overall the structure and function. To understand how a protein gets its final shape or conformation, we need to understand the four levels of protein structure, that is primary, secondary, tertiary and quaternary. So first we are going to discuss primary structure. The simplest of protein structure. Primary structure is the simplest sequence of the amino acids in polypeptide chain. The hormone insulin has two polypeptide chain, A and B. The sequence of the A chain and the sequence of the B chain can be considered as an example for primary structure. So in the primary structure, these are the two important chains. The first one is chain A and chain B. Here the chain A and chain B are the insulin has two polypeptide chains. Okay. The next is secondary structure. Secondary structure refer to the local folded structure that form within a polypeptide due to interaction between atoms. So the most common type of secondary structures are the alpha helix and beta plated sheet. So both structure are held in shape by hydrogen bonds, which forms between the carbonyl oxygen of one amino acid and the amino hydrogen of other. See in the secondary structure, the most alpha helix and beta sheeted plated sheet, both structure are held shape by the hydrogen bonds. So the bond between carbonyl oxygen, that see in the structure beta plated sheet carbonyl oxygen group and amino acids of the hydrogen group. So these are the secondary structure. Next one is tertiary structure. The overall three-dimensional structure of the polypeptide is called a tertiary structure. The tertiary structure is preliminarily due to interaction between the R group of the amino acids that make up the proton. Important to tertiary structure, a hydrophobic interaction in which amino acids with nonpolar hydrophobic R group cluster together on the side of the proton, leaving hydrophilic amino acids on the outside to interact with surrounding water molecule. Also, disulfide bonds, covalent linkage between the sulfur containing side chain and cysteines are much stronger than other type of bonds that contribute to tertiary structure. See in the tertiary structure. Next is quaternary structure. When multiple polypeptide chain subunits come together, then the protein attains its quaternary structure. An example for quaternary structure is hemoglobin. The hemoglobin carries oxygen in the blood and is made up of 4 subunits, 2 each of the alpha and beta types. Denaturation and protein folding. So what is the denaturation and protein folding? Each protein has its own unique shape. Uh, for example, primary structure and secondary structure, tertiary and quaternary. Each one has own unique shape. So if the temperature of pH of proteins environment is changed, or if it is exposed to chemicals, this interaction may be disturbed, causing the proton to lose its three-dimensional structure and turn back into an unstructured string of amino acids. When proteins loses its higher order structure but not its primary sequence, it is said to be denatured. So what is the meaning of denature? The protein lost its higher order structure but not its primary sequence. That is called denatured. Denatured proteins are usually non-functional. So these are the denaturation of proteins. So thank you, students.